seat. We want to start to be able to have the group picture after the session. So you all have a bag on your chair. So thanks, Dorothy, to have that organized. Dorothy was extremely disappointed not to have them before. So thanks again, Dorothy, to have that organized so that we could have them before the end of the meeting. There are only 100 of them, so only one per person. So please only take one, and leave the others for your fellows. Okay, so we will start this new session and leave the floor to Andrey Shkoporov. Thank you. Phage-host interaction in the gut system. Andrey, the floor is yours.
Is my microphone working? Yeah. Okay. Hello, everyone. My name is Andrey Shkoporov. Very few people in the audience know me because I'm not really a structural biologist or a biophysicist. I'm a microbiologist, and I'm interested in the roles that bacteriophages play in complex microbial communities such as the human gut microbiome. So, as we all know, bacteriophages are the most numerous biological entities on our planet, and every environment colonized by bacteria also contain bacteriophages, very often outnumbering the counts of their bacterial prey, as demonstrated in this example with concentrated seawater. And the gastrointestinal tract of mammals is not an exception. Dense and diverse communities of bacteria that inhabit it support equally dense and diverse communities of bacteriophages. So last year we published this quantitative metagenomic study of viral communities in gastrointestinal tracts of two species of mammals, pigs and macaques. This is as close as we could get to the humans. And we found that free viral particles were present in all segments of the gastrointestinal tract, starting from the oral cavity, and all the way down to the distal colon, where the concentrations of free virions reached 10 to the 10 particles per gram of contents. And what's interesting, we also saw, well, lower levels, but still significant levels of viruses embedded into the mucosal layer, and we saw considerable levels of viruses present in parenchymal organs linked with the gastrointestinal tract, such as spleen and other parenchymal organs. So most of these viruses are actually bacteriophages. More than 90% are bacteriophages. And up until just a few years ago, the vast majority of these bacteriophages were completely novel. And if you did metagenomic sequencing on them, up to 99% of reads could not be mapped to any entries in the sequence databases. This situation changed considerably in the last couple of years, when large sequencing surveys were completed for the human gut virome in particular, and people managed to assemble and catalog hundreds of thousands of individual viral genomes, which represented up to 50,000 novel phage species. And with that information, we are now capable of mapping about 50% of reads originating from the gut virome to some entries in the databases. However, if you look at this collector's curve, it is very far from saturation, actually. And we can compare this to the number of known viral species in ICTV taxonomy, which is just under 13,000 species right now. So clearly, the global diversity and this is including phages, viruses of archaea, plant viruses, animal viruses, so all of them. And only in the human gut we have more than 50,000 phage species, perhaps hundreds of thousands of different species. So what are all these different species? A lot of them belong to novel families of tailed bacteriophages, such as these Flanders viridae and Quimby viridae, but one group of particular interest is a novel order called Crossviralis. And one of the representatives of this novel order is what's called crossphage, or we call it now prototypical crossphage. It's the most abundant virus in the human gut ecosystem, present in more than 60% of human populations around the globe, and in many cases reaching up to 90% of the total gut virome and up to 20% of the total fecal DNA, which is, of course, shockingly high levels. And most of these novel phages were predicted to infect bacteria of the genus Bacteroides. It's strictly anaerobic gram-negative bacteria, which are the most dominant group of bacteria in the human gut, so it's not surprising that the most abundant groups of viruses also infect these most abundant groups of bacteria. So currently, the order Crossviralis includes 73 species. Most of them are completely uncultured. A couple of isolates from two different clades were isolated in our lab. One of them behaves like a normal phage on its host, Bacteroides intestinalis, and the other one, isolated using Bacteroides alignin solvents, cannot produce plaques, so we had to isolate it through serial enrichments. Well, sadly, the most interesting representative of this group, for the typical crossphage, was never isolated in culture. Okay, so through this amazing collaboration with Oliver and Fred from the University of York, well, they were able to solve the structure of one of the phages, Cas001, and this actually enabled us through using homologies to annotate many of the previously unknown genes in the genomes of Crossviralis. And although in the order Crossviralis there is the level of homology is not very high between different clades, you can see that the genomes bear certain conserved traits, such as all of them have approximately the same size of 100 kb. They have this typical structure with early, middle, and late transcriptional units, and they have one of the hallmark features is very large genes coding for giant subunits of virion-associated RNA polymerases. Some of them can be up to 6,000 amino acids long. Okay, so one of the interesting features of Crossviralis or cross-like phages is not only their high level of abundance in the human gut, sometimes reaching up to 90% of the virome, but also very high stability of these viral populations over time. In this longitudinal study in humans, where we monitored virome composition in 10 individuals during a 1 year period, we noticed that some cross-like phages were able to persist at very stable levels for the entire duration of observation, and in one person, same phages, same strains of phages were present for more than 2 years in the gut. Okay, so we became very curious as to how it is possible that phages are capable of such long-term persistence, and we decided to replicate this in a model. First, we used germ-free mice, which we monocolonized with Bacteroides intestinalis and also with phage class 001, and we saw the same kind of behavior. Bacterial populations were stable over time, and so were phage populations, and regardless of the presence of phage, there was no difference in the level of colonization by Bacteroides intestinalis. Even though this phage is believed to be strictly lytic, we couldn't detect any identifiable lysogeny modules in it. And no lysogens were isolated also. And on the next step, when we performed this, an in vitro experiment, very simple model with just serial subculturing of the phage-host pair. Again, they reached stable levels, this characteristic virus to microbe ratio of approximately 0.1, which persisted for 23 days. So not only we were able to see this behavior in CRAS-001, but also in another CRAS-like phage called CRAS-002 infecting Bacteroides solvents, and in a phage isolated by our collaborators, we saw the same, and even in a non-CRAS-like phage infecting Parabacteroides dystosonis, we saw the same kind of behavior. And regardless at what level of multiplicity of infection we used to infect, they all reached the same characteristic levels that were characteristic of the of this particular phage-host pair. So then we performed genomic and transcriptomic sequencing of these cultures infected with phage CRAS-001, and we were, of course, expecting to see resistance mutations, point mutations causing resistance, which would accumulate over time in bacteria and maybe counter mutations in phage, leading to the, some sort of an arms race evolutionary dynamics. None of that happened. Instead, what we saw was switching back and forward of the phase variable promoter regions, which preceded 5 operons of Bacteroides intestinalis, coding for 5 different capsa polysaccharides. And that led to suppression of expression of one of them, called PVR9, and activation of expression of several alternative capsa polysaccharides. So when we look at the behavior of the culture infected with CRAS-001, the capsid polysaccharides are initially dominated by this PVR9 type, and gradually after 24 or 48 hours, what happens is that PVR9 is either replaced with a different type, or a second or a third capsid polysaccharide is expressed together with PVR9. And when we looked at the resistant clones, which we isolated, the same thing happened with them. This is our initial wild type. This is a resistant clone which switched expression from PVR9 to PVR7, and then some other resistant clones were combining more than one capsid polysaccharide. But what's interesting, of course, well, because this is phase variation, this is all reversible, so after several passages, this resistant clone switched back to sensitivity, started to express PVR9 once again. And this resistance was also accompanied by visible morphological changes, a switch from a more compact, dense capsule to a more loose kind of hair-like structures, which is associated with PVR7 here. And this phenotype was also reversible, and it led to dramatic reduction of phage adsorption. So when we looked at other phage-host pairs, such as CRAS-002 infecting B. xylene solvents and PDS1 infecting P. dystosonis, the same thing happened with them. There was downregulation of certain capsid polysaccharide loci, upregulation of others. Unfortunately, Bacteroides intestinalis is not genetically accessible, so we couldn't do any knockdowns on it or anything. But Bacteroides tetraiodromicron is genetically accessible, and our collaborators, they did this massive amount of work. They created a panel of mutants, actually, this panel of mutants, in each of which they knocked out each of the capsid polysaccharide loci, each of the 8 capsid polysaccharide loci except for one. So they created a mutant which only expresses CPS3, a mutant which only expresses CPS4, and so on and so on. And what they found out was that if you lock expression of these phase variable capsule polysaccharides to 1, for example, CPS3, you can create a strain which is very sensitive to class-like phages DAC15, DAC17. But if you lock expression into other capsule polysaccharides, they become completely resistant, but simultaneously they become very sensitive to other non-class-like phages, which is, I think, very interesting. And again, this was due to the loss of absorption, at least in the case of DAC15 and DAC17, our class-like phages that we worked with. So naturally, we and the next question, what structures of the phage are responsible for recognition of these capsule polysaccharides? In case of class 001, PVR9 is the capsule polysaccharide that recognizes, and our prime candidates were tail fibers. But despite a very detailed structure that Fred and Oli were able to solve, the tail fiber part, because it's a little bit wobbly, they couldn't solve it in a high resolution. So out of these 5 proteins that we predicted to be subunits, different subunits of tail fibers, and the structure of tail fibers is quite complex. It looks like a cage with 2 layers, 2 types of appendages. We didn't know which ones are connected to which other ones and in what order. So our other collaborator, Christian Cambillot, came to rescue, and he used AlphaFold 2 in order to predict oligomeric structures of all these different possible subunits of tail fibers, and he came to the conclusion that 2 of them, called GP22 and GP23, they are trimeric, and they are likely forming this inner appendages in this cage of tail fibers. However, the interface between them could not be resolved very well, but in the homologous phage, DAC15, he was able to actually predict the structure and the interface between 2 types of trimers as well. And then he did the same for the outer ring of appendages, which were GP25 and GP24, or another combination, GP24 and GP26. We don't know how exactly they are arranged in this outer ring of appendages, but it seems like they're maybe alternating, 2 tip proteins are alternating there. Okay, so very fresh results from the lab. We wanted to check if predictions were correct, and we tried to co-express these subunits in E. coli, and they did indeed co-purify as complexes of predicted molecular weights. And we also tried some binding assays, and they were able to bind to bacteroides cells, but not E. coli cells. But when we co-expressed mismatched pair, GP22 and GP25, there was no complex formation there. So eventually, I want very quickly to show you this very simple model of what we think enables class-like phages to coexist with their bacterial hosts in the gut. This dynamic switching between sensitive phase and resistant phase creates a subpopulation of resistant cells which can survive phage infection, but then these resistant cells, they have to pay the metabolic cost of resistance, or this alternative form of capsule polysaccharide can cause enhanced immune recognition, or else they can acquire sensitivity to a different phage, and this creates an incentive for them to periodically switch back to the original phenotype, and therefore providing this constant supply of sensitive cells that the phage can infect. And as a result, we have this perpetuated coexistence of bacteriophages and their bacterial hosts. But this model doesn't account for some other phenomena that we see, such as possible carrier state. We see that some of the cells are actually surviving phage infection. They can even divide with phage progeny sitting inside of them, but this is very early stages, so I don't want to speculate a lot about that. So with that, I want to thank members of my group involved in this study, my amazing collaborators and my founders. Mostly this study was supported by Wellcome Trust, and I want to thank organizers for giving me this opportunity to talk today. Thank you very much.
Thank you very much, Andrey. Questions? Alan?
I would think that as the strain is changing capsule, that the phages will also be mutating to compensate and be able to infect some of those different types. So did you find mutations in your phage populations, maybe in the genes?
Not on this time scale, not on a time scale that we used in our in vitro and in vivo experiments with, serial cultures or germ-free animals. But on a longer time scale, we do see new mutations appearing in, let's say, tail fibers, in phages. In the human study, we saw that, but it was difficult to actually correlate the significance of those mutations because we didn't know what was going on with the bacterial side.
Super cool to see the different scales from the animal lab and then from the biochemistry. As you said, the phase variation seems to explain quite a lot of it, but as you said, it probably doesn't explain everything. I'm wondering, have you thought about kind of looking at it from sort of a meta-population perspective? So in the mice experiments, I guess you are kind of looking at the whole of the intestine as one, like did you sample from stool or I guess you're not checking different parts of the gut. So I was thinking that if in some places you eradicate the host, this creates like a bacterium-free area of the gut, which can still be colonized then by like kind of classical island meta-population theory.
I agree with you. This was not accounted for in our simple models, but we know from studies from other groups that this anatomical sequestration of phages and bacteria can also contribute towards long-term coexistence because, there are areas, for example, bacteria can get embedded into mucus and become inaccessible for phages, or they can reside in the ducts of intestinal glands, again becoming inaccessible for phages. So we know that this mechanism exists, but there is probably more than one mechanism that contributes towards overall kind of temporal stability of the virion and bacterium in the gut.
So, quick question. So beautiful work. Have you expressed some folded fibers in E. coli without a chaperone, or are you also co-expressing a chaperone?
No, we just expressed the tail fibers and mutants on their own. Do you mean some sort of a dedicated chaperone from the phage? Well, we were not able to locate it in the genome, so we took an easy way. We just expressed the tail fibers themselves. And they are difficult to express, they are difficult to purify, they are tricky, but some percentage of protein apparently folds correctly, and we isolated it.
And do you see superinfection exclusion factors in the carrier state that modify the capsule? Same as, for example, seen in people.
The carrier state is, I just thought I'd mention it, but it's a very early stages. I don't think I am ready to discuss it. We didn't do a lot there. It's just some sort of an observation that might be a carrier state, but I wouldn't, be absolutely 100% sure.
Okay, but do you see superinfection exclusion factors that are capsule specific in your phage genome? Glycosyltransferase.
No, but I'm not sure. Well, you see, they might be there. A lot of the genes are not annotated, really. So there might be something there. it's not something that that was obvious to us.
Okay, thanks. We can discuss later.
You're using germ-free mice or something, germ-free animals for these things, and I was wondering if there was any danger of doing that because the immune systems of those animals are bizarrely different than the immune systems of wild animals. So, in addition to the forces put on these bugs to evolve or to change their coats from the phage predation, you might also have in a natural situation some sort of pressure from the immune system, which might be kind of screwed up in the germ-free animal.
Yeah, obviously this is true. Well, in the germ-free mice, because there is no resident microbiota, there is very little that the local immune system can do against a single bug that you place in it. So they just, they expand, they use the full capacity of what is given to them. So in a natural situation, of course, you have a lot of competition between different species of bacteria. You have this selecting power of local and systemic immune system that, selects against certain bugs in favor of some other bugs. Yeah, well, this is a reductionist model, obviously.
Thank you very much, Andrey. And we could have such a long discussion because the next speaker, so let's thank Andrey. So the next speaker is not here. So we won't know who eats bacteriophages. And so our next speaker is Gordeeva from York, and she will be talking about recognition strategies of type 1 and type 4 BREX systems. Julia, the floor is yours.
Good afternoon, everybody. My name is Julia. I'm currently a postdoc at Applied Artisans Group, but today I'm going to tell you a bit about my master and PhD thesis work. So as this conference is more about structural viruses, I'm not going to go deep into details. And I just focus on a question, how immune system BREX recognizes between host and the virus. Here is the main characters of this research. It's me, my PhD supervisor, Professor Konstantin Virinov, immune system BREX, which is depicted here as a dog because BREX is like a Rex, which is the name of a dog. This system helps Escherichia coli to survive during phage lambda infection. This study began with publishing of the first paper about BREX system. A BREX system, it's an immune system from prokaryotes. This name came from bacteriophage exclusion. It has nothing to do with British Brexit. The core gene of this system is predicted alkaline phosphatase BREX-Z, which is in green here. And the second core gene is putative ATPase, which is C here. And depending on composition of all other BREX proteins, BREX systems were divided into 6 types. And the most widely distributed type is type 1. And the first BREX system was BREX system type 1 from Bacillus carriers. It was a publication from Rotem Thorax Lab. BREX system from bacillus carriers consists of 6 genes with some strange predicted activities. They showed that this system protects from phages, bacillus cells. They also showed that there is no accumulation of viral DNA inside BREX cells. And also they found that this system methylates adenines at these non-palindromic motifs. So we decided to test another BREX system, and we chose E. coli as our model, and we found BREX system in a natural strain of E. coli. It was Escherichia coli HS strain. This system consists of 6 genes with predicted activities like ATPase, DNA methyltransferase, alkaline phosphatase, and protease, and 2 genes with unknown function. We just cloned this system into a low copy number plasmid and a natural promoter and transform this plasmid into a laboratory strain of E. coli. So we decided to choose phage lambda to test BREX defense activity. So as you may know that phage lambda is a temperate phage that can undergo the lytic pathway with production of viral particles. But also this phage can undergo the lysogenic pathway where phage integrates its DNA inside the host and just passively replicates in it. This state is called prophage, and cells that carry out this prophage called lysogens. So you can also induce this prophage from its sleeping state by prophage induction. In our case, we chose a temperature-sensitive strain of phage lambda, and we were able to change temperature and control the lysogenic or lytic pathway of this phage. First of all, we wanted to answer the question if BREX inhibits any of this pathway for phage lambda. What we did was just simply performed efficiency of plaque formation assay, where we prepared clones of cells with BREX system or control cells without, just with antivector, and titrated phage on these 2 clones and compared the titer. As you may see, there is a 100-fold difference between titer of the same phage on these 2 clones, and it means that only 1% of phages were able to escape BREX defense. So we also tested the lysogenic pathway. For this, we just calculated the number of lysogens that we had after phage lambda infection, and we concluded that BREX inhibits both the lysogenic and lytic pathways. Then we decided to design another experiment. So where we firstly incorporate phage inside Escherichia coli cell, and then transform these lysogens with our BREX system. And we noted that after induction, we got very strange phages. So phages induced from cells that carried out our BREX plasmid could infect BREX plus cells in both lytic and lysogenic pathways at the same level as the control cells. So, and when we decided to check what happened to this phage, we sequenced this phage in order to find any mutations or modifications. And we found that BREX system marks DNA of Escherichia coli with methyl groups at specific sites, and also the same modification was found in the genome of Phage lambda induced from BREX plus lysogens. So it was a modification of adenines, these non-palindromic motifs. So we concluded that BREX system methylates adenine, and if there is this mark in DNA of Phage lambda, this phage can easily infect BREX plus cells. But if there is no such modification in phages, such phages couldn't infect our BREX cells. So then we just wanted to answer the question, which BREX gene participates in BREX modification and BREX defense. For this, we just deleted each BREX gene and tested BREX methylation state and defense. So we concluded that for BREX genes, BREX B with a known function, BREX C, predicted ATPase, predicted methyltransferase and alkylase phosphatase participate in BREX modification. So modifying module plus the last gene participate in BREX defense. So we concluded that BREX marks host DNA with the methyl groups and distinguish host DNA from viral DNA by this modification. But if phage acquired this modification, it becomes invisible for BREX defense and can kill this bacteria. Then we decided to test BREX system from another type. It was BREX system type 5. So BREX system type 5 we found only in Archaea, and we choose Archaea Halarcula hispanica. The interesting thing about this system that it has two methyltransferase genes, X1 and X2. So we also created different strains that had partial deletion of BREX genes, complete deletion of BREX genes, and deleted each BREX gene one by one and tested BREX activity. We concluded that BREX system from this Archaea protects cells from 3 of 8 tested phages, and we took one of these phages for further experiment. We just simply infected our culture with the virus and tracked the number of living cells and production of viral particles inside BREX plus and BREX minus cells. So we found out that in BREX plus culture there is no production of viral particles compared to control cells. We also checked genomes of all strains that we created in order to find modifications. So we found modification of cytosines at this CTAG motif, which was present in all strains that we created, which means that it is not mediated by BREX system. It may be some domestic modification, like it happens in Escherichia coli with the down methyltransferase, which is necessary for replication, or it can be some restriction modification system. But what we found that there are two different motifs that were modified in BREX plus strain, but not in control cells without BREX system. So curiously that when we deleted one of methyltransferase, one of such of this motif became unmodified. And when we deleted the other methyltransferase, the second motif became unmodified. And we concluded that each BREX gene methylates its own BREX motif. So then we just calculated the number of BREX genes in all viruses that we tested. We grouped these viruses into several groups. The first group is viruses without BREX system in their genome, and as a consequence, this virus became invisible for BREX defense, and there is no defense against such viruses. Another group was viruses with BREX sites, and as a consequence, with BREX defense. And the third group was virus with lots of BREX sites, but without any BREX defense. And the next question that I want to answer, why this phage escapes BREX defense. And I hope I will share my results next time. So we concluded that BREX type 1 and BREX type 5 work in the same manner by methylation of host DNA and recognition and somehow elimination of viral unmethylated DNA. And in conclusion, so I just compare these two types. So BREX type 1 methylates adenines, while BREX type 5 methylates cytosines. Recognition strategy look like recognition strategy of restriction modification system, but set of BREX genes is quite strange for restriction modification system. There is no production of viral particles for both systems, and also one of my colleagues found anti-BREX protein for BREX type 1 system, which is OCR gene encoded by T7 phage and is known as the anti-restriction protein. For BREX type 5, we haven't found any anti-BREX protein, but I hope I will find. So I would like to thank my PhD supervisor, Professor Konstantin Sverina, for possibility to do this research. I would like to thank scientific popular journal for scholars, which is called Schrödinger's Cat. They created and published these wonderful comics about my research. I would like to thank Professor Fritz Adson for possibility to continue my BREX research at University of York as a postdoc. So thank you for your attention.
Thank you very much, Julia. Indeed, the comics is interesting. Questions for Julia in the audience?
Very, very interesting. The comics were well, obviously. So when you make the phage BREX resistant and you get the methylated DNA in the phage, and then when the infection begins, do you think that the daughter genomes are methylated similarly by the BREX?
Yeah.
Okay, so the BREX system doesn't know.
Yeah, it's just methylated as a host DNA, and just BREX system cannot understand that it's something dangerous for this system, that it's viral DNA. Yeah, it just do nothing.
I noticed they all have a protease. It doesn't seem to have much to do with DNA. Do you have any speculation on what that's about?
So actually we tested all predicted activities for these genes, but we just found some ATPase activity for ATPase and for proteins, but not any alkylase phosphatase activity, no any protease activity. Probably this proteins just works together and show activity when they are just together mixed. I don't know, but we also compared protease domain with all other protease, and it seems it's not protease because the distance between lysine and serine, which is necessary for cleavage, the distance is not optimal for these two amino acids for cleavage.
Thank you, Julia. Other question?
That was a really great talk, thank you. I was curious, I know that you said that you're still potentially looking for some inhibitors and that OCR has already been found in inhibiting a different BREX system. There's also ORB-A that inhibits a different BREX system as well. Do you see any candidates in your phages that seem similar to OCR or ORB-A?
Not really. Actually, my colleague tested different anti-restriction protein, and most of them didn't inhibit BREX system, and we actually do not know why this happens.
Thank you. If there are no further questions, let's move on to the next speaker. Thank you again. So our next speaker is William Robbins from Harvard Medical School, and he'll be talking about cell density-dependent death triggered by viral palindromic DNA sequences. William?
Well, thank you. So, yeah, I shortened the title. I apologize. You had it's sort of long. So I abbreviated this to death triggered by viral palindromic DNA sequences. I don't have any comics. That's a hard act to follow. That was a great talk. And I don't have any movies, so but there's some structure here. So I'm beginning with this slide and just state the obvious, and that is that antiphage, antiplasmid systems have enabled and transformed molecular biology as we do it today, certainly with restriction and modification enzymes, and more recently CRISPR. And in both cases, and as with many other antiphage, antiplasmid elements, you have restriction endonucleases or RNA, DNA nucleases, and in either case you have a mechanism of self-protection or self-immunity. So this is a very seminal paper. This was actually mentioned in the last talk as well. Rodem Sorik has done some bioinformatics and some predictions of new antiphage, antiplasmid elements, as well as those that have been discovered and studied for decades. He named many of these after protective gods. These are protecting the cell from foreign invasion. But these are present in all bacteria. Often more than one is present. And in all cases, these are ubiquitous. They're across bacteria. They're across archaea. Certainly there are innate systems that resemble these in eukaryotes as well. But they must recognize a non-self or foreign signal, it could be DNA, RNA, protein, or some other molecular signal. It acts directly on the signal or is activated by the signal, and then it eliminates either the foreign entity and or kills the host and prevents that foreign entity from replicating or producing. It can employ host factors, and some defense coordination systems actually appear to correlate together and probably coordinate together in some context. So this is a well-studied system. Again, this one is decades old, but this is T7 F exclusion, and if you grow T7, you typically grow this on a female cell, because if you grow it on a male cell, you typically don't recover any plaques. At least it's a very poor efficiency. And the reason why is as T7 infects, here at the bottom, of course you have DNA replication, you have viral DNA or protein synthesis and assembly, but there's signals produced by T7. One is due to the gene GTPase inhibitor 1.2. This is probably changing nucleotide pools. This could be one signal, as well as capsid protein interacts with both a factor from the plasmid as well as a factor from host, F-exclusion A, and this kills the cell and no phage are produced unless you have mutations in 1.2 and or capsid. Okay, so this has been studied again for decades, but the key concept here is exclusion, or defense, whether it's for plasmids or for phages. Often, it always requires a signal, but can be both from the accessory element, in this case PIP-A, but also factors in the host can participate. Okay, so we work on cholera. I'm not going to talk about the pathogenicity or the virulence today, but this is our primary mode of study in the lab. But we are very interested in phages and in the evolution of this pathogen. And we were cloning a simple reporter construct that had LAC-C. This is commonly done for blue-white screens and probably something most of you did as an undergraduate. But what I'm showing you on the, let's see if the laser pointer works. Oh, there it is. Okay. So we transformed this vector. It's got LAC-C, nothing else fancy, but these cells were dying, and these are called blue ghosts. This is actually a paper from John Beckwith in 1981. But the phenomenon here is these cells are growing as a colony and then dying and releasing an enzyme that's chewing up the substrate here. And normally this is what we would see. So there was something about this plasmid, and we decided to dig in a little bit. And we termed this cell density dependent death because we could transform these cells, we could grow them in liquid, they were fine, even up to stationary phase. But if we plated these on a solid surface and grew these, what I'm showing you here on the left here is at high density plating, all of these cells die. These do start to grow as colonies at lower density, but as they grow up to colonies, these also die. But this provided a very nice genetic selection for survivors because very few, you might plate 10 to the 6 here and recover 4 or 5 cells. So it was actually a pretty strong selection. This is the plasma that we were using, and what I'm noting here, what's important is this is LAC-Z. These pink regions are where the deletions were identified in the plasmid, and we found a common region in this deletion that is actually, this is one of these plasmids, it's kind of a hodgepodge. I don't know why it's in there, but there is a T7 RNA3 site. So we found this element that's within this small region that's shared. So we thought this actually might be a signal for what was causing cell death in our Vibrio strain. Okay, so coming back to protective gods in Rotem Sorek, this is one of the systems he identified. It's called the Masao. It's named after an ancient Assyrian god from about 3,000 years ago. But most of our mutations fell within this cluster. There's been some previous work done on this system in Vibrio cholera as an antiphage, antiplasmid element. So that was a little bit disappointing that we weren't the first to find this. We also identified a mutation of LEX-O. This is important in quorum sensing. We think this is expressed at high density, so this made sense. We found mutations in PCMB. This is important in plasmid copy number. This would reduce the signal present in the cell. This made sense. We also identified a mutation in pre-A, which can affect plasmid copy number for some replicons, but is also involved in the host and replication restart in DNA repair. And this is actually important later. Okay, so BSB2 is one of the accessory genomic islands that's unique to the current 7th pandemic. So there are 7 recognized pandemics from the 1960s to current is the 7th. All the 7 pandemic strains uniquely have this island. And if you look at the left-hand side here, this is where the DDM ABC operon, and virtually 95% of mutations were in these 3 genes. Okay, so we don't have structures on this yet. That's something that we aim to do, but I did do AlphaFold prediction on these 3. Pretty good confidence. And what we found immediately is the DDMA, and this has been published as well, has a nuclease domain. And this is similar actually to a nuclease effector in another antiphage system called CBAS. It's similar to other ABC 3-component systems. Some of these are antiphage, some of these have other roles. But DDMB is not important. This is a small globular protein that we believe is just part of the complex. And DDMC is a protein that resembles a very well-conserved large group of proteins called SMC Rad50-like proteins. If you'll note, there's 2 head domains that either into the gene here. There's a coil-coil domain, And most of our mutations were actually within these head domains. There's a Walker A and Walker B motif predicted. And this is just a cartoon structure of how we believe this may be acting. This could be a DNA or an RNA signal, but you have this assembly of this DDMC with DDMB and then the effector protein. And when this recognizes a signal, we're proposing that this DDMA is then activated. Okay, so these are other SMC-like proteins. Includes RAD50, SBCC, and bacteria. If you study bacterial or eukaryotic DNA repair, you probably know quite a bit about this. But you can see the resemblance here between RAD50, SBCC, and this is a segregation protein in yeast. Again, you have these two globular head domains with ATP binding motifs, these long foil domains. And in our case, there's actually a kink, and this might actually be important in binding both DDMB as well as DDMC. So we had a plasmid, we had a putative signal. So we looked at phages. We have a lot of vibrio-phages in our lab. We screened 48. There have not been phages that were significantly restricted by the system in Vibrio shown before. But in this case, we did find 2, and we were really encouraged that the difference in efficiency of plating is almost 10 to the 9th if you plate on a wild type versus a deletion of DDM-ABC. So we had two phages, very well restricted. One was called IN4. We didn't want to use this because there's an E. coli phage IN4, but it's a typing phage from the mid-1960s, as well as a phage that was contaminating a sample that was sent from Bangladesh to our lab. So we had 2 phages to work with. Both are short. We would have called these photophages a couple years ago. They are photophages, but they're in separate subfamilies. One is very T7-like, and the other one is actually a kind of a bizarre group that these phages have 2 RNA polymerases, slightly larger genomes, but they structurally look very much the same. Okay, so we had this mutation in pre-A, and initially we thought this might just be a plasmid copy number suppression or reduction, but we found this actually abolished phage restriction for these 2 phages. These phages don't need pre-A to grow, as far as we know. That's not something that's in the T7-like family. But pre-amines in E. coli and other bacteria are known to be defective in different processes, including homologous recombination, double-stranded break repair, stable DNA replication. And you can knock out pre-A, but you require a couple additional suppression mutations in E. coli. These are really sick. We couldn't do this in Vibrio, and right now we're just stuck with this allele. Okay, so pre-A is known to be important for other phages, as well as some plasmid replicons. In Phage-174, it actually binds this small hairpin site called POTS. This is actually important later. But pre-A binding can recruit other proteins that are then important for replication of this phage. It's actually essential for Phage growth. And if you look at pre-A, where our mutation is located, there is a structure for pre-A in E. coli. This is what I'm showing here. There's a DNA binding domain. There's 2 helicase domains, and there's a small CRR domain. This is where our mutation is located. This is a beta hairpin. Don't confuse this with the DNA or the signal hairpin, but this hairpin structure in the protein is actually important in binding where double-stranded DNA meets single-stranded DNA. So the fact that it's right adjacent to this important motif here, we think this is probably just a pre-A knockdown mutation, and it's not binding DNA as well, but we're currently mutating this region to further explore the role of pre-A. Okay, so as far as the phage, we know these phages are restricted, and we had a signal in E. coli, so we shotgun cloned the phage DNA from these 2 phages. We put this in a plasmid that was permissive for growth. We looked for the same phenotype, cells that died high density, and typically these were 2 to 400 base pair, sometimes bigger fragments of DNA, and we found the same thing. Nearly all the clones that were toxic, it was only about 1 to 2%, had structures that were predicted to form hairpins. I know it's easy to form hairpins when you throw DNA, RNA into a prediction program, but a lot of these had very strong predictive values. We also found the same RNA-3-like element that we originally discovered in our plasmid from T7. This is analogous. This is in that T7-like phage that grows on Vibrio, so this was encouraging. To further investigate this, this is the original T7 sequence. This is the sequence that's present in the plasmid. We took this sequence and we mutated 2 residues to abolish the hairpin. We cloned this in a forward and reverse orientation. And what's important here is here's the original sequence. It's toxic. Sequence 2 is toxic. Reverse orientation is toxic in a DDM ABC dependent manner. But if you mutate 2 base pairs, this is after a couple hours of growth, you recover about 100-fold more CFUs. And this is consistent with what we see in the DDM ABC mutant, regardless of the sequence. We wondered if this was abortive infection. So this is a pulse-filled gel electrophoresis where we did a long incubation with this toxic sequence, C2. And it's kind of hard to see here, but in DDM ABC, looks like we're getting chromosomal degradation, and you don't get this degradation in a mutant. So we think this is nuclease is targeting the plasmid or the phage, but also the chromosome as well. Okay, so our current model is we have some sort of hairpin or some sort of signal or some sort of stalled replication fork where pre-A may come in and bind and then help recruit or coordinate with DDM ABC, activate the nuclease. This is, again, the effector. And this either chews up the foreign DNA or then leads to abortive infection. So, we thought about the role of pre-A. We looked at some DNA damaging agents as well as UV exposure. And what I'm showing you here is wild-type cells across a dose of UV exposure, this is joules per centimeter square, are much more sensitive to UV than a delta DDM ABC, a pre-A, this allele, or the double mutant. So this actually suggests in Vibrio you can have this, you can be anti-phage, you can protect against foreign DNA invasion, but this is going to make you more vulnerable to DNA damage, and this is also true with mitomycin C and some other DNA damaging agents. I just don't have the time to show that today. So our current model is plasmid replication processes, phage replication processes, or DNA damage and repair processes can bring in notably pre-A or other proteins, activate DDM ABC, and lead to abortive infection and DNA damage. So the question still is this a DNA signal, an RNA signal, as an RNA-3 site, or both? You could think about replication. If a single-stranded DNA, an RNA-3 site could still, or a sequence could still form in DNA. We don't know that yet, but we've been playing with E. coli extracts. Remember, we had the signal in T7, and here what I'm taking is extracts of E. coli and extracts of cholera, plus or minus these systems, growing this up to high density, pelleting the PBS, making a lysate. We have an automated French press, and then using these extracts. And here what I'm showing you is T7 DNA in the absence of an extract. If I add the E. coli extract, I lose maybe a little bit, but I'm actually getting some phage replication here, and I'll show you that in a minute. But if you add the Vibrio extract, it has DDM, ABC, and pre-A. This is completely digested. Okay, 5 minutes. And then if you use a mutant here, you maintain your DNA, and if you have the mutant and the allele, the same thing. In these, over the course of an hour, we actually produce phage. This is all done in vitro using extracts. And here, just with the E. coli extract, it's 10 to the 4th. Once we add the extract from Vibrio cholera here with these two systems here, 0 phage recovery, and then again, these mutants, it's back up to 10 to the 4th. So now we can go in and we can inhibit transcription, we can remove RNA polymerase, and we can actually determine whether this is a DNA or RNA signal. And this is actually where we are now. Okay, so these are the 2 papers that we just previously published on DDM ABC. They didn't have a phage that was so well restricted. But what was nice about both of these papers, and coming back to the beginning of the talk, is they found that DDM ABC was largely dependent on another system, which they call DDM-EF, and this can actually bind and degrade plasmid. So here you're actually having DNA damage, and this is where DDM-ABC might be coming in and becoming activated. And in a separate paper, they found that DDM-ABC was largely dependent on another set of antiphage genes in another island that is actually a deoxycytidine deaminase that depletes nucleotide pools. This could stall replication forks. So in this case, or in both these cases, DDM-ABC may be acting in tandem with other antiphage elements that induce damage and then produce the signal that leads to cell death. Okay, so I'd like to acknowledge our lab. I had help with Jonita Tosca and Brad Meter. They were both terrified to work with phage. They had not worked with phage before. I said it would not ruin the rest of their experiments, but we've gotten really back into phage and some of these mechanisms in terms of defense. Of course, John Mathlanos for being very supportive, funding the Harvard OTU cluster for research with AlphaFold 2. So, thank you.
Thank you very much, Phil.
Thanks, Cécile. And just for you and any other future speakers, stop embellishing T7. It's going to take Ian from bearable to insufferable. So just ease on it. But good question. DDMA, the AlphaFold prediction almost looked like a Turbinese large subunit. It almost looked like your nuclease domain, kind of dark, kind of tired, but it almost looked like an ATPase domain on the other half of it. Am I crazy, or?
Yeah, I'll have to look closely at that. It is kind of dark. I apologize. I don't know. I mean, it's interesting that most of our mutations we picked up were actually outside the effector domain, and the idea is that this domain, when this binds, this may fold a little differently and then activate the nuclease. But no, I'll have to look closely and maybe some hidden Markov models and such and see. But thank you. Uh-oh.
Just a quick couple of clarifications. Did I hear you right when you said that pre-A mutants were UP resistant?
Okay, they're not entirely pre-A resistant. They are less sensitive than wild type. And this is in the context of there being an antiphage defense system. But no, even in the absence, yes, less so than wild type. And yes, that goes against the canonical.
Well, yeah, I mean, the coli 3A mutants are UV sensitive. The other thing is another clarification. Does cholera have an RNase 3? And if so, does it recognize the same sequence structure as coli RNase 3?
As far as the structure, I don't know. It does have an RNase 3, but based on our TN-seq sequencing, it looks like it's essential. Otherwise, it would knock it out. But I do have, because I have the E. coli lysate system working, I can use an E. coli that's RNase 3 minus, and that's the idea.
I have a question about the SMC-like fold. I didn't understand mechanistic how you interpret why we need to have a coenzyme fold to act on these. This is used to structure the bacterial chromosome or so.
I'm sorry again?
The SMC proteins, they normally structure the nucleoid and the chromosome of the bacterium. So how mechanistically you could explain how we get in this system? I understand the interest of the nuclease, but not of the SMC.
So the SMCs some of these are segregation proteins and bind the chromosome, and you're absolutely correct. But in bacterial systems like SBCC, if there's a stalled replication port and there's a hairpin that forms, there's work that actually shows that they'll come in and recognize that hairpin and help coordinate with the repair. So these SMC proteins play a lot of roles, and segregation is just one of these. But we believe that it's recognizing, those two head domains are recognizing some context of DNA, probably at replication forks or open complexes. But the pre-A may serve this role too. So there may be some specificity with pre-A. But the fact that there's two head domains, structurally, I mean, you run this through HHRED or some of these prediction programs. All the closest matches are all the SMC proteins, but it's primarily because there's two head domains and then some of that alpha helices. But mechanistically, I can't say, how this is acting yet. But if we get a structure, the hope is to get a structure and we have the complex pulled down now. But get a structure with the signal, and then maybe we'll have a better idea.
Thank you. I remind the participants that there's a prize for those who ask questions during talks. So please ask questions. Don't be afraid. Nobody will eat you. Under 30. No more questions, Bill? Then let's come again. And next talk is from Karen Maxwell from the University of Toronto. She'll be talking about inhibition of virion assembly in antiviral defense systems.
All right. So I'm a bit of a contrarian. I come to an assembly meeting and I'm going to talk about inhibition of assembly, anti-assembly. Am I going? Oh. All right. So most of the talks here have actually been focused on, viral assembly, the lytic part of the pathway. In my lab, we're really interested in studying temperate phages and how lysogeny actually affects bacterial physiology. Prophages, of course, have been shown to encode genes that are important in human diseases. They can transmit antibiotic resistance genes. Most bacterial toxins are transmitted by prophages. And prophages also play a role in antiphage defense. And we're starting to characterize how prophages help to protect the bacterial cell that they reside in from further phage infection. And of course this makes sense because once the phage is integrated into the genome and wants to be maintained as a prophage, anything it can do to help to increase survival of its bacterial host, by extension, increases its own survival. There have been a number of different ways that prophages have been shown to provide phage defense to the bacterial cell it resides in. They've been shown to encode genes that are involved in receptor masking, so incoming phages can no longer interact with the receptor they need to infect. They encode superinfection exclusion proteins, so proteins that are integrated into the bacterial cell envelope, and again, prevent phages in the surrounding environment from infecting the bacterial cell. Prophages have also been shown to encode defenses that act intracellularly. So they've been shown to have CRISPR-Cas systems, or sometimes they just carry CRISPR RNAs and actually hijack the host cell CRISPR-Cas systems to provide defense, and also a number of different gene silencing methods. Repressors are one example of that, but other HNS-like proteins have also been shown to be carried by prophages and used to silence incoming phages. So again, in our group, we've been studying this group of closely related Pseudomonas aeruginosa-phages, and in a study we did I think it was about 10 years ago now, we looked at genes of unknown function in this group of Pseudomonas aeruginosa-phages. What you're looking at here is 5 different Pseudomonas aeruginosa-phages, and there's something flying around back there. And every arrow is an open reading frame. The arrows that are colored dark gray, we know the function of those. Most of them are involved in replication of the phage or virion assembly. Genes that are colored light gray are conserved among this group of phages, but they're genes of unknown function. And then the colored genes are genes that are highly variable between these prophages. And we were really interested in how this variable genes that are found in these prophages, what are they doing for the prophages and how are they affecting the bacterial cell? So Joe Gandhi-Denemy, when he was a student in Alan's lab, cloned all of these genes that are shown in different colors. Genes that are colored the same color are homologs of one another, so these peak genes are all the same. And what we found was that Prophages often encode genes that provide further defense against phage infection. And they do this by a number of different mechanisms. So there's pilus modifying proteins like Christina talked about earlier this morning that provide phage defense. But also we found some that are involved in intracellular resistance against phage infection. And I'm going to talk to you today about these two genes that are found here in phage JBD26 and how they provide defense against further phage infection. So GP31 is the gene that we discovered. It's a little 83 residue protein. It's encoded between the small and the large terminase in phage JBD26. And when Joe cloned this gene into a plasmid and expressed it in Pseudomonas strain PA14, what he found was that it gave very strong resistance to phage infection. We wanted to confirm, actually, that this phage infection, or this phage resistance mechanism was actually active from the prophage state. So what we did was we went in and we created a deletion in this gene 31 and showed that we have phage resistance here, the wild-type JBD26 lysogen, and then when we knock out GP31, it restores the ability of this phage to plate. And so that showed us both that this gene was providing phage resistance, but it was being constitutively expressed from this lysogen, or being expressed from the lysogen, and was at high enough levels to provide pretty strong phage resistance. And I'll just point out here that this gene is in the opposite orientation to the morphogenetic genes, and we have this predicted promoter upstream of this gene, and that is driving the expression of this gene from the prophage. So we knew from a plating assay, looking at plaques on solid media, that we had this very strong phage resistance phenotype. We were interested to see what this kind of resistance looked like in liquid growth. We were wondering, could we see, is this an abortive infection mechanism or is something else going on? So at low MOI of 0.1, you're just following the optical density of PA14 growth, and when we infect with phage at about the 3-hour time point, and of course, you'll see the phages eventually take over the culture and lyse all of the cells. Now, when these cells are expressing GP31, we see this very strong phage resistance. So the cells are no longer, the phages are no longer to overtake the cells. Now, when we actually look at high MOI, so an MOI of 10, the bacteria cells are greatly outnumbered. What we saw surprised us a little bit. And what happens is that it actually looks like the phage lytic cycle is proceeding at the same rate in the presence of GP31 and in the absence of GP31. And that's not what you see for an abortive infection mechanism. Abortive infection mechanisms, as soon as the cells are infected, they die. And so this told us that we were looking at something that was maybe different than a typical abortive infection kind of system. The next thing we looked at was what kind of particle intermediates were being produced. So could we see any production of phage intermediates? And what we found was that here's a wild-type infection. We see normal-looking phages. In the presence of GP31, what we see are these empty heads and no tails. Now, we were kind of surprised by this. It looked to us like maybe two parts of the pathway, the assembly pathway, were being affected. So normally, something that blocks head assembly, at least with lambda, the typical system we all think about, the head and tails are assembled in separate pathways, of course, so if one pathway is affected, you don't see effects on the other pathway. And so, because we had seen, so these empty heads that we were seeing, we were curious about whether or not DNA was being packaged into them. So what Premal did was he did an infection in the absence of the presence of GP31, and then he subjected that to DNase treatment and looked for protection of the phage genome inside the capsid. And what he saw was that even in the presence of GP31, the DNA was being protected. So that told us that, the replication cycle was proceeding normally, and the DNA was being packaged into the capsid, and yet somehow after that it was falling out. And we think it's just an artifact of the preparation for when we were looking at the negative stain EM. So again, I'll remind you, we see in the presence of GP31, we see these empty heads. Pramal made a terminase mutant, so can't package the DNA inside the head, and what he sees are empty capsids and lots of tails. That's what we expect to see. And he also created a tail tube mutant, and what we found was that actually, if there are no tails attached to phage DMS3, it appears that even though it has all of the genes necessary for head filling and the head-tail connector proteins that are there, the genome is just not stable inside the capsid in the absence of tails. We were also really curious about how phage GBD26 actually prevents self-targeting. As I mentioned, these phages are all very closely related to one another by sequence, and what Pramal noticed was that we had phages that were insensitive, so the two at the top are insensitive to GP31 activity, and the ones at the bottom were sensitive to or subjected to the GP31 activity, so they cannot replicate in the presence of GP31. And same genomic position, the large and small terminases are on the order of greater than 95% sequence identity. What Pramal noticed was that when GP31 was there, phages that were insensitive to it always had this gene 30 upstream, whereas phages that were sensitive to GP31 activity either had a different gene or had no gene in there. So what Pramal did was he tried to generate a mutant lacking GP30, and he tried, and he could get mutants that were missing GP30, but anything he found that was missing GP30 was also lacking GP31. And so I'm showing here the JBD26 mutant lacking both 30 and 31. It plates normally on PA14. It's lost the ability to plate in the presence of GP31. Pramal then took phage DMS3, and he swapped in gene 30, GP30, into DMS3, and he asked, could this now plate in the presence of GP31? Oops. And what he found is that it did. This must be the repair that happened when I uploaded my talk. So oh no, I guess there it is on the bottom. It was already there. So here it is, DMS3 with GP30 in it. We can see that it has now regained the ability to plate. And so this tells us that the GP30 is both necessary and sufficient for inactivation of GP31. And Pramal has gone on to show using bacterial 2 hybrid that GP30 and GP31 directly interact with each other. And so our proposed model of GP31 activity is that during lysogeny, the JBD26 prophage is constitutively expressing GP30. If a competing phage, a related phage comes in, it actually is inhibiting tail assembly, and we haven't figured out exactly how it's inhibiting tail assembly, but we have some good ideas, again, because these phages are so closely related. Most of their tail genes share, or tail proteins share a very high sequence identity, and so really the primary suspect right now is the tape measure protein, and we think that GP31 may be binding to the tape measure protein and inhibiting assembly of tails. When JBD26 actually wants to enter the lytic cycle, because GP30 is in line here with the terminase genes, as soon as it starts expressing the late gene operon, GP30 is expressed, it binds up all of the GP31 that's in the cell, and JBD26 is able to replicate normally. Now, I think this brings up a couple of interesting things about anti-phage defense, and actually Bill set me up for this quite well. So in this system, we don't think there's a trigger. We think that GP30 is just being constitutively expressed. It doesn't affect GP26 itself because it has the antidote that it needs. And the other thing is that this actually is abortive infection, but it's not the cell killing itself. The cell dies, but it's passively using the phage-lytic cycle to actually cause that cell death. And so I think when we're thinking about anti-phage systems in the bigger context, that I think we don't have to think, is there a trigger that's going to upregulate the system, or does the cell or the defense system actually have to have something that does the killing? So again, phages have figured out how to do something that the bacterial cell has bigger, more complicated systems to deal with, and it's done it with one tiny little 83 residue protein. Now, when we look at the same genomic position in a lot of other Pseudomonas phages, and these are not so closely related, but if we look between the large and the small terminase, what we find is there are a lot of genes there. So this is a really common place for an insertion. We don't really know why. It doesn't really seem to make sense that it would be a good spot for the phage to integrate sort of random genes. Kamal has cloned a few of them, and he's shown at least 2 others act in viral defense, and we are continuing to characterize other genes that we find in this region. And then again, coming back to this bigger picture, when we look at across phage genomes, even with these small phage genomes, we have 61 genes in this. More than half of them are still of unknown function. I think there could be a lot of other anti-phage defenses in there. Phages that have bigger genomes, hundreds of genes, I think there's an enormous mass of untapped antiviral defenses out there, and we are continuing to investigate just a small area of them. So I'd like to just thank the people that did the work. Most of this project was led by a grad student in my lab, Kamal Patel, with some help from other people, and of course Joe Bondi-Denemy, who started out isolating these phages and cloning these initial genes. And I'm happy to take questions.
Thank you very much, Karen. Again, young ones. Sorry.
Really great talk, thank you. So my question is, do you have any predicted structures for any of those little tiny proteins that you have? Are there similarities that you see between some of those different proteins that you have?
Yeah, we have done structure prediction on some of them. Typically they don't look like anything, or they'll be like a little 3-helix bundle. We don't really get, the ones we've looked at, and we have looked at quite a few, we're not getting any good information about what they may be. And most of them don't hit anything of predicted function even.
Okay, sort of a second question, if I can. So you said that you saw these stalled procapsids, you said empty heads.
Yeah.
But they looked mostly like procapsids, not necessarily expanded capsids. So do you see any interactions also with the large terminates at all?
No, so that was, when we first, I would argue they don't look like empty capsids. They're thinner, and if you look at the terminase mutant here, these look like procapsids. These things have collapsed down things. So these actually look like what we saw for Hong Kong 97 heads that were lacking the head-tail joining protein, where we know the DNA is packaged and then falls out. I don't know, Bob's shaking his head. So that was, I think there probably are some proheads. That was our initial thing. It's right between the terminases. Of course, it's interacting with the terminases, and it's not.
If your DNA is coming out, how is your DNA's protection assay show you that you have protection if you're losing it then also?
I think we're losing it during I think we're losing it during processing. I think it's something, maybe the staining, it's pretty rough conditions that you put it through. So I think our DNase protection assays show us in the cell, it's being packaged, it's being protected, and I think that, you know, the tail mutant having these empty things, because we were really surprised that we were seeing these empty capsids. We were sure it must be something to do with packaging. Yeah.
It looks like you're cleaning up the extract a lot before you do the EM.
We are, because the micrographs were so super ugly that I was even embarrassed to show these ones. Yeah, well, it doesn't look pretty when you do that. There's a lot of garbage around.
Karen, excellent talk as usual. The position between of 30 and 31 between terminases is freaky. There's something going on. But during the lytic cycle, when you see the expression of 30, do you see repression of 31? Because it's trying to be transcribed in the opposite direction.
Yeah,
you have a mechanism of suppressing that activity so you don't get
we haven't actually looked directly at that, but I think we probably are, and because there was just a paper that came out that was talking about toxin, antitoxin, or something, and yeah, and it's something that we're thinking that we're going to look at, yeah.
A very nice talk, thank you. And so your non-county goes operon is on the late gene of the phage. Do you think if you move that operon to the early genes, it will change something for the blocking? And also, if you change the direction for the GP, for example, GP31, do you think the block is still like exist? And maybe the third question is, probably if you put both infection result and induction result together, and you will have some insight on how this system works. I think, yeah, probably.
So your first question, I think if we moved both genes to an early region, that we wouldn't see. I mean, I think we'd still see the inhibition because we'd still have expression of 31, but I don't think the phage would be able to overcome it anymore. I think it's key that it has GP30 there to turn off 31 activity at the right time. I can't. What was your next question?
If you put the GP31 in another direction, like the same.
Oh yeah, I don't think the direction of that matters that much. Although that gets back to Paul's question, that, you know, is it important to have it there in the backwards direction? Because as soon as you start blasting through the morphogenetic region, it's probably going to downregulate expression from that reverse strand. So that could be an important regulatory component of it.
Thank you.
Okay, great. I guess you don't exclude the chaperones as targets, but my question, you have to tell chaperones, but my question is if you can have any strategy to get escape mutants that will allow you to find out.
Great question. We tried super hard to get escape mutants and couldn't. Like, Pramal spent months and months and months. So what we're doing now is starting to make hybrid phages, regions where there's differences in the tail proteins, to see if we can bypass it that way. I think it might just be that, you know, it's easy to get a single mutant that can escape. I think if you need multiple mutations, that then becomes really hard to isolate escape mutants. So for one of these other antiphage systems, he actually has isolated escape mutants for it, but this one we haven't been able to. Yeah.
Such a small protein too.
I know.
Intriguing. Okay, if there are no further questions, thank Karen again.